Hi everyone, my name is Morin. I am a student in biology at University Laval, and today I'm going to present to you the results of our master's degree in a presentation called A Study of Apis mellifera Hygienic Behaviors to Fight Varroa Destructor in Quebec and Ontario. This project was made with the support of the CRESA, the Centre de Recherche en Sciences Animales of Deschambault, and also the Ontario Ministry of Agriculture, Food and Rural Affairs. If you have any question, I have put my email address so you can send them to me afterwards. So, before we enter the main subject of my research, I'm going to touch a little on Varroa Destructor. As you may know, this parasite is one of the main causes of death worldwide in apiculture, due mainly to its transmission of viruses and its exponential growth in the colony. So the Varroa has two phases. First, it spends some time feeding on the adult bees, and then it enters brood cells to reproduce. So the Varroa mite will be trapped within the brood cell with the larva, and when it will be capped by the workers, it will start laying eggs that will mate with each other, and at the end of the cycle of development of the bee, up to 2 new Varroa mites will emerge with their mother. So because this cycle is only taking a few days, and that the colony is growing exponentially during the summer, so is the population of Varroa. And as the population of bees is decreasing before the winter, this is a moment where it is the most critical for the colony and where the Varroa is able to cause the most damage. So there are many treatments that are available today, and we are currently dependent on them. But there is a resistance, we are seeing arriving, and we are trying to develop new ways to fight this parasite. As an example, we can look at natural behaviors that some bees are able to do by themselves. So for example, there are the hygienic behaviors, which are complicated behaviors that can be divided in two main steps. So first, the worker bee is going to detect an odor that will be emitted from the brood that is either diseased, injured, or dead. And then the second step is that the worker or another worker is going to open the cell and remove the dead brood or the infected or whatever. And this behavior is going to decrease the contagion between the different individuals, and is going to reduce, in the end, the presence of the pathogen in the colony. And what is very interesting for our Varroa problem is that we are seeing a hygienic behavior that is specific to the Varroa. So the worker is able to detect a Varroa mite that is reproducing in the cell, and is going to disturb it by either removing the pupa or opening the cell, like you can see here. And what is really great about that is that these are traits that are heritable. So that means that it can be transmitted from generation to generation, and we can do artificial selection on them. So the behavior that is specific to Varroa Destructor is called Varroa Sensitive Hygiene, or VSH. So as we just saw, the worker is sensing the presence of the Varroa under the cell cap, going to chew on the cap and open the cell. And what is really great about this is that we, as researchers, are able to find traces of this behavior happening. So as you can see in this picture right over there, this is the underneath the cell cap, and this mate trace here is an indication that the cell was open and then closed back. And then the worker is going to remove the infected pupa. There is another interesting behavior that can be also found in the some colonies of bees, which is called suppressed mite reproduction, or SMR. The SMR is the failure of a mite to reproduce effectively, so it will not be able to produce at least one adult mated female at the end of its cycle. So that's slowing the growth of the Varroa population when there are many Varroa that are not able to reproduce. So our goal is to do artificial selection of these traits, but selection in bees is very complicated. So what the bee does is a result of both the environment, its genetics, and the epigenetics. And we know that the genetics of the bee is very complicated because of its way of reproducing. So there is one bee per colony that is mated by many different drones, and all the different workers in the colony do not have the same relationship with one another. So if they have the same father, they are more closely related than if they don't have the same drone father. As well, the bees cannot be kept in a controlled environment, so you cannot put them in a staple. You have to they have to go fetch their food in the environment. They are at the service of the different weather. So this is causing great pain for the breeders that are trying to identify the bees that are expressing the most VSH, because we do not know, in fact, what is the factor of the environment in this behavior. And the VSH and SMR were discovered not very long ago, not even 20 years ago. So there is a lot to learn about them before we really understand the mechanisms. So in this context, I fixed myself 2 objectives for my master's degree. The first one was to compare the performances of selected colonies in different environments. So as we will see, I had 2 different lines of bees in 2 different environments. And my first hypothesis was that the Varroa resistance that is induced by the VSH trait would be similar in different colonies developing in the different environments. And my second hypothesis was that the performance traits would also be similar. And the second objective is that I wanted to clarify the links between different traits that are associated with the VSH and SMR and other hygienic behaviors. So the hypothesis that I decided to test was that some traits would permit us to predict the SMR of a given colony. So now I'm going to present to you what I did to test this hypothesis and what were my results. So first we're going to look at the performance comparisons of the selected colonies in different environments. So everything started in the summer of 2018, where we bred new queens for our project. So there were 2 main lines of bees. So we had one VSH line and one hygienic line, and we produced 26 and 24 nuclei that we shipped to Ontario, and we kept 14 and 16 nuclei in Deschambault, so in the province of Quebec. And for the other part of my project, I also added 2 lines with 12 and 14 nuclei. So the main phase of the project was in 2019, and the test and the monitoring happened that summer. And the colonies were dispersed in one apiary in Ontario and 5 different apiaries for the province of Quebec. So here, it's not really that important to focus on each different number, but I just wanted to show you how the number of colonies evolved during the summer of the test. So at the beginning of the summer for the first test, we already had some losses due to wintering, and there was some lack of food in the environment in the spring. So we started off with about 12 colonies in Quebec and about 18, 17 in Ontario. And due to mainly swarming and presence of diseases, it evolved through the season. But we did not have too many losses, and the analysis were able to be made like this. So to know how the different environments differed, I characterized the temperature in each apiary. Then through melissophanology, we were able to identify the different flower species that the bees were going to collect the nectar. So this is a technique that was done by Melissa Girard, and she analyzed the pollen in the honey sample and was able to know which floral species were in the areas. And then we made a satellite cartography that was done by Claude Four to know what were the difference in cover that was melliferous or non-melliferous. So these are for the apiaries in Quebec. We have the 4 apiaries represented in yellow, and around them is the surface in a 5 kilometers radius, and in pink is the covered by melliferous plants. So it will be interesting to compare with the one in Ontario, but sadly I do not have the data yet. Hopefully we will have it soon. For the floral diversity, here each column is an apiary. So the first one here is the apiary in Ontario and each one of the apiaries in Quebec. So this is a list of the different species that were identified in each one, in each sample. And the names in red combined account for 90% of the abundance in each yard. So this is just a quick portrait of what we can find. So we can see that there were many more species in the Ontario yard than there were, for example, in this one in Quebec. So to test my first hypothesis, that was that the traits are associated with the Varroa resistance would differ or not between the different environments. We tested 3 things. So first we analyzed the Varroa population on the bees, so in the phoretic phase with the bee samples. Then we made an assessment of the hygienic behavior with a free-scale brood test. And finally, we tested for the SMR. So we do this by opening cells until we find 35 Varroa females, and then we analyze what the family is like, so we can say if she was able to reproduce correctly or not. For a second hypothesis, so we measured the honey production of each colony by weighing all their honey supers. Then we did the brood spring growth. So we did two inspections in spring, where we basically measured the surface of brood of each frame, and then we were able to convert that in number of cells total in the colony. So we did that at the mid-May and mid-June, and we were able to see the growth between the two. And finally, we measured fall strength, so the measurement of number of frames that were covered by bees on top and bottom of the box just before wintering. So that gives us an average of the bees that were present and the strength of the colony. For the statistical analysis, so we did two-way ANOVAs so we had 4 groups, so 2 lines and 2 environments. So this is my lines in Ontario, the VSH and the Hygienic, and the same in Quebec, the VSH and the Hygienic. So this is the result for the Varroa population. At the end of the season, you can see the Varroa mite infestation levels, so the number of mites per 100 bees that were found in the samples. And we could not see a significant difference in the infestation levels. Now for the hygienic behavior, we have the percentage of clean cells after 24 hours. So we can see that there is a significant difference here, that the VSH line, in fact, was more hygienic on average in the Quebec yard than in the Ontario yards. So we're going to talk a little bit about this later, but this could indicate that something in the environment influenced the hygienic behavior. For the SMR, I do not have the data for Ontario yet, but I can show you that the averages of the colonies that were developing in Quebec are about 30–35% of non-reproduction, and no difference between the two lines. But we'll see for the environment later. For the honey production, so there is great variation between colonies, but no significant difference between the environments. If we look at the spring brood development, so we can see each group was identical or similar at the beginning of the spring, at about 10,000 cells of brood in the colony, and they developed through the spring, and we cannot see a significant difference in the end. So all groups, we can say that they develop similarly. For the false strength, there is a difference. So we can see that the colonies in Quebec are the bees in these colonies covering more frames, so we could see that they are stronger. Though when we look at the data and the conditions of the test, we could see that there was a huge difference of temperature when the readings were done. So it was about 18 degrees colder for the Ontario colonies, and we know that there is a relationship between the temperature and so the colder the temperature, the smaller is going the ball of bees is going to be smaller. So we cannot really say that it is because of the environment, but rather most likely that they were just more tightly in Ontario. So if we look at this summary, we're similar in Varroa population. We cannot say yet for the SMR, and there seems to be an effect of the environment on the hygienic behavior. And there was no difference for the honey production, brood spring development, and we really cannot say for the fall strain itself. Now we're going to look at the method and the results of my second chapter, which was to clarify the links between VSH SMR and other variables. So here is just a table that shows number of colonies of each line here in each of my 5 apiaries. So we had a total of 35 colonies for this experiment. Okay. So first, we did 2 hygienic behavior measures. So we did the classic freeze kill, which we use liquid nitrogen to freeze the brood, and then 24 hours later we can see the percentage of clean cells in the brood. We also did a pin test, which is mostly done in Europe to compare. It's the same principle except that we use an entomological pin to pierce and kill the pupa, and the readings are done after 9 hours. Here is a diagram that shows the VSH SMR method. So to test for VSH, you need to have a highly infested brood frame, so highly infested in Varroa. To acquire that, you need to have a yard that is to breed Varroa, basically. So what you want to have is untreated colonies that are isolated in a yard, and you're going to use these cages first to have homogeneous laying of eggs on a frame. So you're going to trap your queen on it because you want the larvae to all have the same age. Then you're going to put back the frame with eggs in the Varroa donor colony until the frame is capped. So when you have a capped brood, you have Varroa inside. So you're going to take out the brood frame, do an initial evaluation where you're going to count the number of Varroa to have a percentage of infestation. Then you're going to take this frame and incubate it in the colony you want to test for VSH. So during this time, if the colony has the VSH trait, it's going to work on the brood, remove some of the Varroa, and when you take it out, you can see the difference in infestation. So before we go on, we're going to look a little bit closer at the initial and final evaluation. So at the initial evaluation, this is where you have your brood frame with capped brood. And something we develop is that we can separate the frame in 2. So each donor colony can give us 2 frames to test 2 colonies, which is a great innovation, if we can say, because it is very complicated to have this yard and have the good infestation levels. So yeah, when you have your half brood frame, we open 150 cells to count Varroa infestation before the testing, so a percentage of infestation. Then, before putting it in the test colonies, we take a picture to be able to count the number of capped brood cells on the frame. Then it goes 7 days in the test colonies, and at the end, we take it out, and we're going to take another picture to see the difference in the number of brood cells. So we are able to see how many or what proportion of the pupae were removed during the testing. Second, we are going to do another evaluation of the infestation rate so that we can see the difference, and this is going to be our VSH our VSH trait. At the same time we do this, we're going to look at each individual cap, wax cap, and we're going to see, like we saw in the beginning of the presentation, if the cap was uncapped and then recapped by the workers. So we're going to have a recapping rate. And finally, we're going to find 35 Varroa mites to see if they were able to reproduce correctly or not, to have the SMR level. So what analysis did I do with all of this? I used all my traits that I presented to you earlier and tried to see if we could predict with one of these the SMR level of a colony. So for example, if I only take the measure of our population growth, will I be able to predict if it's going to be highly SMR or low SMR? And this is what we found. So indeed, we saw that the population growth of the Varroa in the colony was a great indicator of the SMR level. And this is the relationship between SMR and population growth. So here is the SMR, and here the Varroa population growth. And what we see is that the quicker the population of Varroa grows in the colony, and the lower is the chance that the colony has a high SMR. So in other words, if the population of Varroa doesn't grow a lot during the summer, there's a high chance that it has high SMR. And another trait that was significantly able to predict SMR is the recapping activity, total recapping of the cell. And the higher the recapping activity, the higher the SMR was. And we could not see a relationship with the other traits. So we just saw the relationship of the traits with SMR. So now is the relationship between each other. So we have the pin test with the free scale and all the others. And I want to put your attention on these. These are the correlation levels. So the closer it is to 1, the stronger the relationship between them. And here it shows that the total recapping activity is correlated to the recapping of only the infested cells. So it shows that the more they recap in general, and the more they are going to recap the infested cells. And also it is correlated to VSH, which is super interesting. So in conclusion, my results show that the environment plays a significant role in hygienic behaviors. So we had 2 lines, 2 genetic lines, and they had different scores in different environments. That doesn't mean that it's going to be always the same. Example, all the colonies in Quebec are going to be more hygienic than in Ontario. That doesn't mean that. It just means that when we compare different colonies that are not evolving in the same environments and we test for hygienic, we have to be aware that the difference we see is not necessarily due to the genetics when we are doing breeding programs. Also, what we found is that the recapping activity and the Varroa population growth during season are linked to SMR. So that supports the hypothesis that it's the workers' activity that is causing the non-reproduction of mites. Also, hygienic behavior that was evaluated with free-scale brood tests is not related to SMR and VSH. So that is suggesting that it's not really that when we test for hygienic with free scale, we are not directly selecting for VSH. So we have to do a different kind of test for that. And finally, our results show that the recapping activity is correlated to VSH and SMR. So this could be an interesting trait that is easier to evaluate than the SMR VSH themselves, and to include this in a breeding program that could increase the resistance to Varroa. Thank you for listening to my presentation. Even though my English is a little bit broken, I'm sorry about that. I want to thank especially Kelsey and Les from the Ontario Tank Transfer team and all they did together with their team for the data collection. I also want to thank the CRESAT team for the help with the test and the management of the colonies in Quebec. And my dearest colleagues, Mireille and Sigolène, for their help during the summer. So please ask me your question via my email. I will also try to send a PDF of this presentation if you want to review the slides without having to see the video all over again. Thank you.